Hello and welcome to Bio40 Pre-Lecture 22. Today we're going to discuss cytogenetics. Cytogenetics is the study of wild-type and mutant chromosomes. Up until now, when we've talked about mutations, we've mostly dealt with base substitutions and small insertions and deletions. But with cytogenetics, we're going to talk about large-scale chromosomal changes. We mentioned aneuploidy a while back. This is having too many or too few copies of a given chromosome. There are also structural abnormalities, rearrangements within and between chromosomes. This will be our focus for the next couple lectures. Chromosomal abnormalities are observed in about 0.6% of live births. But that vastly underestimates how common they are, because such conceptions often fail to make it to term. And chromosomal abnormalities are not simply a problem in germline cells. Aneuploidy and rearrangements are found in nearly all cancer cells. Though interestingly, there is some debate over the degree to which they are a cause or an effect of cancer. However, chromosomal rearrangements are not always bad things. They have historically been a driving force in evolution. You may have heard figures about humans and mice sharing about 85% of their DNA. But if you map the locations of genes from the 19 mouse autosomes to where their human homologs can be found, you'll see there's been quite a lot of shuffling over the 80 million years since we've had a common ancestor. You can even see some major changes between us and our closest ape relatives. Chimps, gorillas, and orangutans have 24 pairs of chromosomes rather than our 23. But careful studies have revealed that this is because sometime in our past, two chromosomes joined together and became one. On human chromosome 2, we see the remnants of interior telomere sequences, and a second centromere that is no longer functional. There are four main types of chromosomal rearrangements that we're going to cover: deletions, duplications, inversions, and translocations. We will focus on their causes, their physical characteristics, and their genetic implications. These are depictions of the four types of chromosomal rearrangements I just described. Take this chromosome that has been divided into 8 regions, A to H. In a deletion, regions of the chromosome are just lost. In a duplication, a region of the chromosome has been copied and now appears twice. With an inversion, no information is added or lost, but the order of the genes along the chromosome changes. While deletions, duplications, and inversions are all changes within a chromosome, translocations are rearrangements between chromosomes. Translocations are very similar to crossovers, which we've seen before. But rather than being between homologous chromosomes, we have an exchange of information between non-homologous chromosomes. All of these rearrangements can be the result of incorrect repair of multiple double-strand breaks. If there is a break between regions A and B and between C and D, but A is wrongly attached to D, we can get a deletion of B and C. If instead, A is attached to C and B to D, nothing is lost, but the order is changed, an inversion. Now suppose there are two breaks in homologous chromosomes, between B and C of one chromosome and C and D of the other. If the pieces are swapped during repair, you can get a deletion in one chromosome and a duplication in the other. The same sort of event between non-homologous chromosomes can lead to translocations. Improper crossing over can also cause each type of rearrangement. We've talked about crossing over occurring between homologous chromosomes, but crossing over can actually occur between any homologous sequences of DNA. And there are sequences called simple sequence repeats and transposable elements that are repeated all throughout our genome. Suppose there are homologous sequences between regions A and B and between C and D. Depending on how they're oriented, crossing over could cause a deletion, or it could lead to an inversion. And crossing over between different regions of homologous chromosomes can cause both insertions and deletions. Finally, homologous sequences can be found on non-homologous chromosomes, and crossovers between these sequences can lead to translocations. Many of these rearrangements have severe consequences. Being homozygous for a deletion is frequently, but not always, lethal. It depends on the size of the deletions and the genes affected. Of course, deletions that include essential genes will be lethal. But even if no individual gene in a deletion is essential, the loss of many non-essential genes can add up to a lethal effect. Even heterozygosity for deletions can be detrimental. This is for the same reason we talked about monosomy being a problem. Even when you have a single working copy of each gene, you don't have the right dosage. When heterozygosity is not lethal, there can still be interesting phenotypes to study. For example, take the interesting shape of a Drosophila wing from a mutant heterozygous for a deletion that includes the gene Notch. We talked about how smaller deletions were important for Benzer's work, mapping mutations within a gene. But larger deletions can likewise be used to help map genes along a chromosome. Deletions can uncover recessive mutations. Much like how the phenotype of a recessive allele of an X-linked gene is always visible in males, deletions can cause stretches of hemizygosity. A normally recessive allele appears dominant when paired with a deletion. This is sometimes called pseudo-dominance. So if you pair a recessive allele with a deletion and see the recessive phenotype, you know the deletion overlaps that gene. Take this example here. If the recessive scarlet trait is visible when paired with deletions 3 and 4, but not 1, 2, or 5, the scarlet gene must be located in the region where 3 and 4 overlap with each other, but not any other deletion. This information can help tell us where a gene is physically located on a chromosome. This is because large deletions can be seen cytologically. When homologous chromosomes pair together, and one has a deletion, the part of the chromosome that no longer has a homologous region to align with loops out. When there are only 2 strands of DNA, this can be hard to see. But in certain cells, flies have these odd polytene chromosomes that make visualization possible. Specifically, they are found in the salivary glands of the larvae. In these cells, the chromosomes undergo 10 rounds of replication without separating, leading to over 1,000 sister chromatids. And with the homologous chromosomes combined, that's over 2,000 strands of DNA. With this many strands, it's certainly possible to see chromosomal rearrangements. Here's an actual image of a stretch of a polytene chromosome. The key to seeing rearrangements is the banding pattern. Normal Drosophila chromosomes always have the same banding pattern. And you can see here that along most of this chromosome, the bands stretch across all 2,000 plus strands. But there's a region marked with arrows where the pattern is off. The strands on the right are missing a series of bands that are present on the strands on the left. The chromosome on the right has a deletion covering the region of those bands. The banding pattern in Drosophila is well characterized, with each band given a specific number. This allows us to assign mutations to certain bands. Take the gene white here. When a recessive white mutation is paired with these 3 deletions, the white phenotype appears. This is not the case when the white mutation is paired with these 2 deletions. So the white gene is located where the first 3 deletions overlap with each other, but not with the last 2 deletions. The only chromosomal band that fits this pattern is band 2 of region 3C, so the white gene must be a part of that band. Similar logic tells us that the facet gene is part of band 7, and the roughest gene could be part of either band 5 or 6. Now let's move on to duplications. These can be classified as tandem or non-tandem. In tandem duplications, the copied segment immediately follows the original, while in non-tandem duplications, the copy is dispersed to another part of the chromosome. In either case, the copy can be in the same order as the original, or the reverse order. Duplications often do not have phenotypic effects. No information is lost, there are just extra copies of certain genes. However, just as we compare deletions to monosomy, we can compare duplications to trisomy. Having the extra dose of gene products can lead to different phenotypes. Take the Notch gene again. We already saw that being heterozygous for a deletion can lead to these wing notches. And now we can see that a duplication of the gene leads to extra wing veins forming. Also, some effects are the result of the new sequences formed at the borders of repeats. This can be seen in flies with a bar eye phenotype. The number of facets that make up the eye are reduced, and the eye takes on this odd shape. The phenotype is caused by the duplication of the 16A region of the X chromosome. Normally, the 16A region is between the 15F and 16B regions. But the duplication creates an abnormal junction where the end of the 16A region is followed by the start of another 16A. This was the first evidence that the location of a gene along a chromosome can affect how it's expressed. We'll learn more about this when we get to gene regulation. Bar-eye flies can also help us illustrate another problem with duplications: their inherent instability. For homozygous duplications, if the duplicated regions misalign before crossing over, the recombinant chromosomes can gain or lose a copy of the region. Females that are homozygous for this duplication would often have wild-type offspring, as well as offspring with the more severe double-bar phenotype, the result of having 3 tandem repeats of the 16A region. Organisms that are heterozygous for insertions have an interesting issue during prophase 1. There are multiple ways the homologous chromosomes could align to maximize a pairing of homologous regions. There are two different ways that just have one of the duplicated segments looping out. A crossover in these cases will not change anything. But you could also have a situation where the duplicated regions on the same chromosome align with each other. A crossover in this case can lead to the duplication being deleted, another way that duplications are inherently unstable. So to summarize, large-scale deletions and duplications alter many genes at a time, so even heterozygotes can display phenotypes. This is because the effects are additive. Perhaps a deletion or duplication of a single gene would have no effect, but deleting or duplicating a region of several genes can add up to a noticeable phenotype. Deletions and duplications can also alter a gene's relative location along a chromosome, which can affect its expression. Finally, it's important to note that duplications are a driving force in evolution. When a gene has an important role, mutations that change its function will be detrimental. But if that gene is duplicated, one copy can maintain its normal role, and the second is free to accumulate new mutations and develop novel functions. Let's cut it off there for today. Next time, we'll finish our discussion of cytogenetics with inversions, translocations, and aneuploidy.